Hey guys, I am Miss Alleman and I'm going to be doing the Turgor Pressure Potato Lab with you guys today. There's a couple things that I need for this experiment, so I'm going to go through each piece of equipment. I have a 500 milliliter beaker of water. I have another beaker with 500 milliliters of a 4% salt solution, so you're also going to need some salt. That's going to be about 1 tablespoon of salt in this one. And then you have a 500 milliliter beaker of water with 20% salt solution, which is about 5 tablespoons of salt. You are also going to need a potato, and I have cut my potato up into little potato sticks, and I have 6 potato sticks total because I'm going to be putting them in my 3 beakers, and so I'm going to have 2 for each beaker just to make sure that my results are accurate. I also have a plate, and I have labeled my plate into 3 different sections. I have water, 4%, and 20%. That way when I'm taking them in and out of the beaker, they have a designated spot and they don't get flip-flopped. I also have tweezers, or in science we call these forceps, and that is helping to get the potatoes out of the water. And then I'm also gonna have a timer, which I'm just gonna use my phone. So before we get started on this experiment, you are going to need to follow along with your lab handout. Has Mr. and Mrs. Potato Head up at the top. Now, for the background information, you need to recall your prior knowledge on functions of the cell wall, functions of the vacuole, and then think back to the notes that you just completed over turgor pressure. So pause the video here and go ahead and answer that background information section, and then we will begin on the lab. Okay, so we have our background information completed, and now we're gonna look at number 6 on your lab sheet, which says, draw a picture of what each potato stick looks like before they are soaked in the different solutions. So I am going to show you a picture of the plate and what the potato sticks look like. And you are going to draw this on your lab sheet. Now, notice that it says on your lab sheet, draw your pictures on the observation page. That's in big bold. So on the very back page, looks like this. This is going to be where you make all of your drawings. So you can see up at the top, this references back to number 6, and you're gonna draw the 3 potato sticks in these boxes. So let's go ahead and make our observations now. Okay, so we should have our observations done for the before soaking it in the solution. I'm going to take my forceps, and for the section of the potato sticks marked water, I'm going to put those 2 in the water beaker. My 4% goes in the 4% beaker, and my 20% goes in the 20% beaker. Then I'm going to take my phone, and I'm going to set a timer for supposed to be 20 to 30 minutes. So I'm gonna set it for 20 minutes, and we will come back in 20 minutes, and if we need to add more time, we will. 20-minute timer has started. So let's sit, wait, and see what happens. Okay, so it has been 20 minutes. So I'm gonna take my forceps, and I'm gonna get the potato out from the water. 4%, and 20%. He wanted to run away. Okay, so for your lab sheet, we are now on number 7. And number 7 says to draw the observations for after. Now what I am going to do is I'm going to show you how flexible that these potato sticks are. And however much that it bends, that is what you are going to be recording and drawing pictures of in the end. So I am first going to just show you how much they bend, and then I will show you a set of pictures, and the pictures will be a better reference as to what you will be drawing. So first with the water, I can feel that it's pretty stiff and it's not going to bend very much. No matter how much pressure I put, there's not a lot of flexibility in this one. In my 4%, I can feel it is a little bit softer. Now, remember, this was just the 4%, so it's kind of supposed to be in the middle of my tap water and my 20%. So when I try and bend this one, it does bend a little bit more. What? No! And that's why we have tweezers, because now my hands are all wet. That's okay. So whenever I'm bending it, it is going to have that little bit of a curve in the side. So what do we think is going to happen with the 20%? 20% definitely, I can tell before I even bend it that it is a lot softer. It is a lot more flexible. So I can see I can bend this and curve it pretty far. So I will show you guys pictures in just a second of how much each potato is curving. So as I went from plain water to 4% to 20%, that curve is going to be more dominant, I should say. Okay, so look at those pictures and then you are going to record it in your lab observation sheet. Okay, so now you have completed the second set of observations, which was after it had been soaked. So you should have the top two completed. And now you are ready to start your conclusion questions, which is the second page of the document. And it talks about which potato stick had the greatest amount of turgor pressure and the least amount of turgor pressure. So when you're looking at which one was flexible versus which one was not, this is a potato, and potatoes are plants. So think about what the cell wall would do if something was turgid and tough or flaccid and flat. So compare that to how much it bends. Salt, the more salt that was added, the more it dehydrated that potato. So if it dehydrates the potato, it's taking water out of the vacuole. So think about that process as you're trying to figure out which one had the greatest and least amount of turgor pressure. Then it says for number 10, you're going to draw a picture of what the potato would look like under the microscope. And you're comparing it to what it looks like in water, which was my stiff, rigid potato, and what it was like in 20%. So this is kind of going to relate back to greatest and least amount of turgor pressure. But the drawing that your teachers want is what it would look like underneath a microscope. So if I put a potato underneath the microscope, I would be seeing individual cells. And remember, we just said that the potato was a plant cell. So you're going to be drawing a plant cell for what the water potato would look like, and then a plant cell for what the 20% potato would look like. So if you need to refer back to your turgor pressure notes that you took earlier in the class, you can do that. Then you have number 11 and 12 saying, if a plant is wilting, explain what's happening. Make sure that you include some of those organelles of the cell wall, the vacuole, and what is happening inside of the cell. And then number 12, is turgor pressure caused by an internal or an external stimulus? And that was one of the first videos that you guys watched today of what something happens inside or outside of an organism. So think about what happened with the potatoes. Think about what organelles are involved, and if this is happening inside of the potato or outside of the potato. And then make sure that you explain your answer for that one. Whenever you are done, then you are going to submit your assignments, whether that is to your teacher in person or in the appropriate location online. And that concludes our turgor pressure potato lab. Super fun.